All right, I think we're ready to get started here. One sort of just administrative thing I mentioned, I think it was a week ago. Let me toss it out one more time. On Tuesday of next week, is the one time you get a chance to take a makeup exam in this class. So if you missed either the first or the second test, and I told you you should take the makeup exam, Tuesday of next week is your shot, right? It's from 12 till 2. The syllabus tells you what room it's in. Don't miss it. We'll all be sad if you do. If you didn't miss the first test or the second test, you got no problems. And then, I think it was a week ago, I asked, just out of some curiosity, I suppose, how many of you thought that you might be here on Tuesday of next week? If you hadn't heard, there's a big holiday coming up. A lot of people leave town, and 9 a.m. or 9:30 a.m. is kind of probably the peak time to get out of Dayton on a Tuesday in anticipation of Thanksgiving. I'll be here, right? I'm going to talk with you. The lecture notes tell you that the next topic is bacterial genetics, I think is what it's called. And I think I gamed the system a little bit. At some point, I kind of fiddled with things. The next logical thing for me to talk with you about after today's lecture is the regulation of gene expression, how genes get turned on and turned off. I've got no better material to work with in the whole class than what I'll tell you about in terms of how genes get turned on and turned off. That's the next logical thing for me to talk with you about. But what I've got in the lecture notes and the PowerPoint presentations is, instead of talking with you about that on Tuesday, when I'm expecting not many people will be here, is I'm going to talk with you about viruses and their genomes and how they store information, right? So if you had to miss a lecture, I'm telling you, that one might not be such a bad one to miss. You can probably get what you need off the Integrity recording. I'm cool with that. I don't want to discourage you from coming. I just don't want you to have any anxiety. I want you to have as happy a Thanksgiving as you possibly could. And if you need to leave on Tuesday, I'm not going to hold that against you. All right? Not in any way. All right? But so we'll have fun talking about how viruses do their things. But I want you to come back after Thanksgiving really excited, because, again, I got nothing better to talk with you about all semester long than what I'll talk with you about on Tuesday, and that's how genes get turned on and turned off. Today, the deal is I'm going to finish talking with you about the central dogma, okay? And again, that sort of sets the framework that I need then to talk with you about how genes get turned on and turned off. The central dogma of molecular biology, very simply, is this: information in living systems, living cells, flows from DNA to RNA to protein. Okay? Good orienting principle. That's going to get you out of trouble a lot in subsequent classes that you'll be taking as biology majors or med students or whatever. That's a good guiding principle. Information flows from DNA to RNA to protein. That is what people call the central dogma of molecular biology. With that said, I know there's some smart alecks amongst you. A few of you have talked to me already. It's like, but wait a minute. Isn't there a chance for information to flow from RNA to DNA? It's like, yeah, sure, there is, right? That's what retroviruses are all about. That's kind of a little asterisk, a little footnote to the central dogma. It is possible for information on occasion in certain circumstances to go against that flow that is the central dogma. So sometimes you can get information out of RNA and get it into DNA and then use it to get RNA in proteins. Fine, okay, well, I'll accept the little footnote and asterisk. But check this out. If you can figure out a way to get information from proteins back to RNA or back to DNA, you got yourself a Nobel Prize, absolutely guaranteed. Just as Watson and Crick were confident and certain that they were going to get a Nobel Prize if they figured out the structure of DNA, if you could crack that problem, guaranteed you got a Nobel Prize. to date, no one has figured out a way, and living things do not seem to have figured out a way to convert information from the sequence of amino acids and proteins back to the kind of information that is stored within DNA. All right, so central dogma, information flows from DNA to RNA to protein. Well, once in a while, maybe from RNA back to DNA. I don't know what's going on with the pictures behind me here, but nonetheless, we'll figure it out. And but we don't have any instances, we don't have any system in nature where we see the information going back from proteins upstream through that central dogma. All right, well, I can see an outline here. And now you guys can see one on the screen as well. So, well, momentarily you could see it on the screen. Huh, curious. We'll see how this works. So, in class on Tuesday, I talked with you. I tried to make a big stink about 5' versus 3', right? And I kind of gesticulated, saying maybe you should go back and take a look at the pictures of the ribose sugar and nitrogenous bases. And it occurred to me, I did a bad thing. I should have had that picture to show you instead of just waving my hands. Here's the picture that I was referring to. Let me fix any damage that I did in lecture on Tuesday by pointing out to you what I mean by 5' and 3'. So what I told you on Tuesday is nucleotides have 3 parts. There's 3 important components to any nucleotide. There's the nitrogenous base, there's the 5-carbon sugar, and then there's the phosphate group. All nucleotides have those 3 things. There are 4 different kinds of nucleotides that we find in DNA and 4 that we find in RNA. What makes them different is which nitrogenous base they have, right? So for DNA, we've got C, T, A, and G. For RNA, we've got C, U, A, and G. All right, so 4 different nitrogenous bases. I've talked with you a lot about base pairing and how they interact with each other through hydrogen bonds. That's all swell. That's not what I want to focus on here today. But you got your nitrogenous base. You got a 5-carbon sugar. Tell you what, that 5-carbon sugar is ribose, okay? So it's a 5-carbon sugar. You can see the 5 carbons in all their glory. There's one there, there, and there. So 1, 2, 3, 4, 5. Notice how the carbons have names. The first one coming around the circle is the 1' carbon. The second one is the 2', 3', 4', 5'. I got interesting stuff to tell you. You know, if you're thinking about what do I need to know about these carbons? What level of understanding do I need to have to help me with the final exam or with other classes that I'll be taking? Here's a little bit of trivia, something interesting about these carbons. The 1' carbon of the ribose sugar is the carbon to which the nitrogenous base gets attached. All right? That's all that's happening with the 1' carbon. It's a space holder of sorts, but its job is to be that's where the nitrogenous base is attached. The 2' carbon has a story. It's an interesting story, and it's what this panel down here is all about. Now, I expect, not because of anything I've said, but because of what people say in elementary school these days, you probably all know DNA is short for deoxyribonucleic acid, right? I think kindergartners hear that these days. It was a big deal when I was in college to know that and be able to impress my parents. That's what DNA stands for, deoxyribonucleic acid. Now everybody seems to know. Jon Stewart knows. I heard him talking about it on TV a couple of years ago. So everybody knows what DNA stands for. But it's deoxyribose. You know, the ribose part makes sense. We're talking about the ribose sugar, but it's deoxyribonucleic acid. The reason DNA is deoxyribo is because in DNA, instead of having a hydroxyl group dangling off of that 2' carbon, there's a hydrogen. Okay, so it's deoxy. D without oxy, the oxygen of the hydroxyl group. it's not there. So on the 2' carbon for deoxyribonucleic acid, there is no hydroxyl group. But if you know that DNA is short for deoxyribonucleic acid, I think there's a good chance you know that RNA is short for ribonucleic acid. You know the difference between RNA and DNA, aside from the uracil thiamine thing? The difference between DNA and RNA is RNA has a hydroxyl group on that 2' carbon. So here is the deoxyribose. Here is the regular ribose. If you want, you could say oxyribose, but that doesn't, nobody bothers to do that. So that's the structural difference between DNA and RNA is what's happening at that 2' carbon. But where the real story, the important stuff is hanging out is with the 3' carbon and the 5' carbon. I got absolutely nothing. I got to do some research, check things out on Wikipedia or something, because I got nothing for you about the 4' carbon. That's the most remarkable thing about the 4' carbon is there's nothing interesting happening with the 4' carbon. But there's important stuff happening with the 5' and the 3' carbon. Okay? The 5' carbon is the one to which a phosphate group gets attached. So the 1' carbon is where you put the nitrogenous base. At the other end, at the 5' carbon, is where you put the phosphate group. And when you link one nucleotide to another nucleotide to go from 2 mononucleotides to 1 dinucleotide, or when you want to make a dinucleotide and a mononucleotide into a trinucleotide, when you want to extend that chain, you do it by making phosphodiester. These oxygen bonds here are, that's an ester bond, that's an ester bond. You do it by making phosphodiester bonds between one nucleotide's 5' carbon and another nucleotide's 3' carbon. And you see how that plays out here, because what you got is that 5' ribose sugar with its nitrogenous base on the 1' carbon. And at the 5' carbon, you got that ball that is the phosphate group. Okay? The 3' carbon is connected by way of a phosphate group to the 5' carbon of the next one down the chain. Why am I talking about 5' and 3' so much? I'm telling you, it's because it's a pretty big deal. It's going to end up being important for you. Take the time. Make sure you wrap your head around 5' and 3'. It translates ultimately, as I said to you on Tuesday, to the difference between the beginning and the end of a DNA or an RNA molecule. And also, it's the functional equivalent of left and right when you're talking about reading text on a page. Okay? When you look at a DNA molecule at the structural level, what you'll see is that at one end of the molecule, there's a 5' carbon with a phosphate group flapping around, dangling in the breeze. Okay? At the other end of the molecule, there will be a 3' carbon with a hydroxyl group just dangling around, flapping in the breeze. If you wanted to make that string of nucleotides one nucleotide longer, you can only do it in one way. You can only add nucleotides on to the 3' end. Okay? You can only bring in a new nucleotide and tack it on down here. You can't do it up here. It's part of this idea of this is the beginning and you proceed to the end. That's the only spot new nucleotides can get added on. Now, my saying that may make some of you think, well, there must be a way you can put a nucleotide on at the 5' end. And organic chemists would certainly say, well, of course you can. You just need to set up certain conditions to do that. But the thing is this, living things don't do that. It may be physically possible to do that, but the enzymes, the DNA polymerases and the RNA polymerases that are making these molecules in our cells, unequivocally, without exception, don't do that. You always add nucleotides onto the 3' end, never to the 5' end. Okay? So DNA synthesis occurs from the 5' end to the 3' end. RNA synthesis occurs from the 5' end to the 3' end. Ribosomes, which I'll be talking with you about in just a little bit today. Ribosomes work from the 5' end of an RNA molecule to the 3' end of an RNA molecule. Everything is 5' to 3'. And don't find yourself thinking that you can slip by somehow without having some appreciation of the difference between the 5' end and the 3' end. It's going to come back and bite you in all sorts of different ways if you're not comfortable with that. So if you're not comfortable with it after what I've just said, not a big deal. It's actually not that hard to wrap your head around. Just stare at this picture for a little bit, come back to it while you're watching the Browns march to victory on after the Thanksgiving feast and such. But, you know, that's all it's going to take. All right, so enough said about that. When I told you about this universal genetic code, right? So on Tuesday, that was a big deal. 64 different ways you could arrange the 4 different nitrogenous bases in sets of 3. All right, when you're looking at them 3 at a time, there's 64 different ways you can combine them. 61 of those 64 combinations communicate to a ribosome to put in a particular amino acid. Okay? UUU tells a ribosome you need a phenylalanine for the next amino acid in the protein that you're synthesizing. UAU tells a ribosome, okay, you're putting in a tyrosine here, right? 2 different amino acids, 2 different triplet codes. 61 of 64 say use this amino acid. 3 of them say nothing. They're effectively communicating to the ribosome there is no amino acid that you put in at this point in the polypeptide, and that's effectively telling the ribosome this is the end of the road. This protein is done. Don't add any more amino acids to this chain. We call those 3 codons stop codons because that's effectively what they tell the ribosome to do. I'll show you some pictures in a little bit, but I'll tell you right now, what's physically happening during translation is the ribosomes come to a stop codon and they sit there waiting for a tRNA molecule to bring an amino acid in for that codon. There is no tRNA for the stop codons. And so the ribosome sits there. And if the ribosome sits there for 30 to 60 seconds or so, it says, ah, I know what happened. It's a stop codon. And it falls apart, walks away, releases the new protein, and lets the protein go off and do its thing. So it waits a certain amount of time. If it fails to get an amino acid for that particular codon, it says this is a stop codon, and off it goes. When there is a tRNA, like for all the rest of these, those get cycled in milliseconds, fractions of a second. So waiting 30 seconds is a pretty long wait to make sure that there's no amino acid that should be put in for these 3 different codons. But there you go. You got this universal genetic code. It's a redundant genetic code in the sense that there are many amino acids for which there are multiple synonymous codons. I slipped in a word just now that I should have used more deliberately in lecture on Tuesday. I said there are amino acids that phenylalanine has 2 synonymous codons. UUU means the same thing as UUC. In English, when you have 2 words that mean the same thing, they're synonyms. 2 codons that tell a ribosome use the same amino acid, we call those synonymous codons. So for phenylalanine, there are 2 synonymous codons. For leucine, there are 6 synonymous codons. In English, 6 words that all mean the same thing. Here, 6 codons that are all telling ribosomes use leucine now. So synonymous codons are the ones that are telling the ribosome use that same amino acid. And then here's a picture I threw at you just to get you a graphic, a way of picturing the principal difference between the central dogma as it plays out in prokaryotes versus eukaryotes. Okay? Central dogma. Information flows from DNA to RNA to protein. In prokaryotes, transcription, the conversion of the information from DNA to RNA, and translation, the conversion of the information from nucleotide sequences to amino acid sequences, transcription and translation in prokaryotes take place at the same place and at the same time. So as an RNA polymerase is making an RNA copy of a gene, ribosomes are right behind that RNA polymerase trying to translate that RNA sequence into a protein. That doesn't happen in eukaryotes. In eukaryotes, instead, what we've got is transcription. It happens exclusively in the domain of the nucleus. RNA polymerases are only functional and doing their thing in the nucleus, whereas ribosomes do translation exclusively in the cytoplasm. So transcription and translation are physically separate, and they happen at separate times in eukaryotes. From the very beginning, when I've talked with you guys about prokaryotes and eukaryotes, I've tried to subliminally, maybe not even that subliminally, kind of blatantly on occasion, tell you when you hear prokaryote, have the word simple come to mind. When you hear eukaryote, complex, I hope, comes to mind. This picture by itself tells you, oh, okay, it's a little more complicated, right? Well, here's the thing. Why would you bother with something complicated as opposed to something simple? Is there an advantage to complexity? I'm trying to think of a good analogy for you right now, right? You know, how about an iPhone 7 with, you know, a camera, a forward-facing camera, back-facing camera, cool speakers, right? You got a lot of stuff going on with that iPhone 7. The original iPhone, did it even have a camera? I can't recall. I suspect it didn't, right? The speakers, I know, were pretty lame. They were lame to the point where you had to invest in external speakers to be able to hear music and think it was worthwhile listening to the music, right? Why do you go for the more complicated thing? The more complicated thing lets you do more stuff, right? It lets you solve more problems. It lets you accomplish more things than the simple things. The simple thing, that's fine, right? It doesn't break as quickly, you know, all that stuff. There's some advantages to simplicity, but there are also some advantages to complexity. Why do you bother with this more complicated central dogma rollout in eukaryotes relative to prokaryotes? It's for that reason. This complexity that we have, the separation of transcription and translation, lets us do a lot more stuff. It lets us actually be much more complicated than we would have been otherwise. And if there's one thing that I can point to as proof of that, and I think I can give you a pretty good list, but the top of the list is the fact that eukaryotes can have single genes code for more than one protein. Beadle and Tatum and Archibald Garrod left us with this idea of one gene, one enzyme, or one gene, one protein, roughly equivalent in their way of thinking. For us, we've got 32,000 some genes in the human genome, and yet we can make 250,000 different proteins during the course of our lifetimes. And if you have a defect in any one of those 250,000 proteins, it's probably going to cause you to either be dead or to have some sort of genetic disorder, right? So the fact that you're not dead, I'm confident in that, looking around the room. The fact that you're not dead and the fact that you don't have any blatant genetic disorders that are jumping out at me as I'm looking at you all tells me that most of your 250,000 proteins are doing just fine, all right? But you can make many more proteins than your genome at a first pass should have allowed you to construct and to make. And the reason that you can do that is because you can process the information in your RNA molecules before ribosomes get around to translating them. And that's pretty much where I left you on Tuesday, was this idea that in eukaryotes, we can edit the information in our RNA molecules. So here's what happens. In a eukaryote, since transcription's happening just in the nucleus and translation's happening just in the cytoplasm, what's happening is the RNA gets made by an RNA polymerase. Okay? And then we call this a pre-mRNA. If you want to get really technical to impress people, you could call it an HN-RNA. It's a heterogeneous RNA. That's like sophomore level talk. So if you want to impress people with how well you're picking up on what's going on in Bio 1120, don't call it a pre-mRNA. Go hardcore. It's an HN-RNA, heterogeneous RNA. But you make a primary transcript. You make an RNA copy of the whole DNA region for this particular gene, and then RNA processing or editing occurs within the nucleus, where you modify the information content of that RNA. Okay? And it's only when all the modifications are complete that we start to call it a messenger RNA, and that messenger RNA then gets exported to the cytoplasm, where it encounters ribosomes for the first time, and translation can begin. In prokaryotes, you go straight from DNA to messenger RNA, and then translation. In eukaryotes, you have this intermediate step that gets tossed in. You get a primary transcript, or a pre-mRNA, or an HN-RNA, all equivalent terms, that gets made into an mRNA. And what I told you on Tuesday, I'm going to tell you again right now, eukaryotes modify their RNA molecules before they're messenger RNAs. We modify them in 3 ways. We change their 5' end. You put on something called a 5' cap, a nucleotide that's not encoded within the sequence of the DNA gets tacked on to the 5' end. Frankly, it's a fairly trivial modification, but it's something we do to a lot of the RNA molecules that are going to need to be translated. So we put another nucleotide on at the 5' end. We tidy up the 3' end, so the RNA polymerase can just keep transcribing away, but an enzyme will come along and cleave off the RNA molecule as it's being transcribed, and tack on a string of a couple hundred A's, adenines, to give you a poly A tail at the 3' end. I'll tell you why that might be functionally important in a couple minutes. And you also modify, in a big way, the information content in the middle of the RNA molecules through a process called splicing. And it's splicing that allows you to have, as a human being, as a eukaryote, it's splicing that allows you to take a single nucleotide sequence and use it to spell out the instructions to make more than one kind of protein. Let me show you how. So, central dogma again, transcription, translation, DNA, RNA, protein. We've done that enough. Here's transcription as it's playing out again. This is just another chance for you to think about 5' and 3'. Notice the RNA molecules getting made in the 5' to 3' direction. The RNA polymerase is actually scrutinizing the template DNA strand in the 3' to 5' direction, but it's making an anti-parallel new RNA in the 5' to 3' direction. So that's usually what we'll say is the synthesis is happening 5' to 3'. It's all about the baby RNA molecule. Here are those first 2 kinds of modifications. If you looked at a primary transcript as it's coming out the butt of an RNA polymerase in a eukaryote, what you would find is that at the very beginning of the RNA molecule, a guanine gets tacked on by this strange triphosphate group linkage. That gets called a 5' cap. So we add a nucleotide onto the 5' end in a funky kind of way. At the other end, you cut off the RNA molecule. And once an enzyme finds this particular string of nucleotides, it's called a polyadenylation signal. That enzyme that recognizes the signal cleaves the RNA molecule at some point a little downstream of the poly A signal and tacks on a couple hundred A's. And in the middle, you'll find the protein coding segment, particularly after splicing has occurred. But I want you just to take a moment, maybe take a step back, and look at the lay of the land of this RNA molecule. This is what a ribosome will see. Okay? This is a finished RNA molecule for a eukaryote. It's a messenger RNA ready for translation. This is what it sees. It sees at the 5' end that funky cap. Cool. It sees a start codon. Notice how they've kept the color the same as what was going on with the genetic code. Subtle thing, right? But you guys know what a start codon is. Do you remember? It's AUG. That particular string of 3 letters, start codon, codes for methionine. That's perhaps the one part of the genetic code that would be reasonable to expect you to remember. That says something specific to a ribosome. That says start translation here. Hey, Mr. Ribosome, I'm a shiny new RNA for you. Translate me, please. The ribosome's going to say, oh, fine, you are a shiny new RNA molecule, and check it out. Here's a start codon. What the ribosome will do is it will latch on at the cap and then move 5' to 3', looking for the first start codon that it encounters, and that's where translation will begin. Okay, so the first AUG that it finds, it says, oh, look at that, start codon. I can do something with that. It brings in the amino acid methionine, and then it's off to the races, going triplet codon by triplet codon, 3 at a time in lockstep, all the rest of the way until it encounters a stop codon. Color has been preserved, apparently, from that previous slide here as well. These are one of those 3 codons for which there is no amino acid. The ribosome gets the message, stop translation here, and the ribosome could really care less about what else is happening over here because it falls apart and goes off in its own separate way. Okay? Doesn't quite fall apart. There are people who will tell you that the ribosome, as it's fallen away, clips off 1 or 2 of those A's from the poly A tail. Takes a few of them off, knocks them off as it goes away. It's sort of like that poly A tail you might think of as sort of a counter that tells you how many times you've translated this messenger RNA molecule. Once you've made a messenger RNA molecule, it wouldn't be good if it stuck around forever. It'd be nice if it had a fixed number of proteins that you make from it. If you want a lot of proteins, maybe you give it an extra long poly A tail. If you want just a couple, a short poly A tail. But it kind of short circuits all of gene regulation if you just keep translating that RNA forever, right? And so they have a built-in, a planned obsolescence, if you will. They're going to ultimately become, the A's will go away. When the A's are gone, other enzymes come along and start chewing up the RNA molecule from 3' to 5', and so they're not around long after the poly A tail is gone. But what I'd like to encourage you to do is just take a step back and look at the lay of the land. There's a stretch of RNA in the messenger RNA that is transcribed. Think what that means. Okay, an RNA copy got made. It is transcribed, but it is not translated. How about that? Ever encounter that in a high school biology class? I bet you nobody ever bothered to draw your attention to that before. In the messenger RNA, there's a region at the beginning, at the 5' end, that is transcribed but not translated. There's an RNA molecule there. It got made from the information in the DNA, but the ribosome doesn't use that information to make a protein. It waits until it finds a start codon before it starts translation. We got a fancy name for that region that is transcribed but not translated and is at the 5' end, that fancy name is the 5' untranslated region. All right? Nobody says 5' untranslated region. They say 5' UTR. Right? That's what the hip, cool molecular biologists will tell you. There's a 5' UTR. What do you suppose is going on with the 5' UTR? If it doesn't serve a purpose, it probably wouldn't be there. There's a lot of regulation that's happening there. In eukaryotes in particular, but prokaryotes as well, making an RNA molecule isn't the last word on the subject of making the protein. Ribosomes get some responsibility in terms of making the decision, should I translate this RNA molecule or not? And a lot of that gets decided based on what the ribosome sees in the 5' UTR. Under some circumstances, the ribosome might come away saying, well, I'm not going to second guess making the RNA, but I can tell you, given the way the circumstances are right now, I don't think it's a good idea to make a protein off of this. And it sniffs out information in the 5' UTR. By the same token, at the 3' end, there's another stretch of sequence that is transcribed but not translated, cleverly named the 3' UTR. Okay? So stuff that comes after the stop codon, but before the polyadenylation signal, is the 3' untranslated region. The long and the short of it is, all that's important or of consequence for most RNA molecules in the 3' UTR is the fact that there's a polyadenylation signal in it that pulls in an enzyme that cuts off the RNA from whatever comes after it and replaces it with a string of A's so that you could have that sort of planned obsolescence, the counting mechanism of the ribosomes doing the business of translation. So there's stuff happening there, but it pretty much boils down to polyadenylation. The stuff between the start codon and the stop codon, tell you what, we got a name for that. That's the coding region. Makes sense, right? That's what's coding for the specific string of amino acids that will make a protein. It codes for that. You'll often hear people refer to the coding region in a different way. Instead of saying it's the coding region, it's synonymous. They'll say it's an open reading frame, an ORF. That's the way people will talk about it, too. It's going to be an ORF. They won't say an ORF. They'll say ever. I've never heard anybody say ORF. I've heard people say ORF. I've heard people say open reading frame. I've heard people say coding region. Those are all roughly the same thing. Why do you suppose they call it an open reading frame? Well, it's an open reading frame because the ribosomes, as they're going 3 at a time through those codons, don't encounter a stop codon. The stop codon is what would close or shut down the reading frame, but it's an open reading frame because it's not interrupted by a stop codon. So from here to here, it's triplet codon after triplet codon that tells ribosomes put an amino acid in here until you get to the stop codon. Then the coding region is over. Then the ORF is over. The open reading frame is done. After that, there's nothing for ribosomes to see any longer. Where I finished on Tuesday was with this slide, or something close to it. And I told you, okay, here, though, in splicing is where the wholesale modification of the information content of our RNAs occurs. You can't do splicing in prokaryotes because transcription and translation are happening at the same place and time. But you can do splicing. You can do this really large-scale modification of the information content of our genes at the RNA level in eukaryotes because transcription's in the nucleus and translation is in the cytoplasm. Okay? What's up with splicing is this, is that even though an RNA polymerase transcribed from here all the way through to here, and maybe beyond, even though an RNA polymerase made an RNA copy of this whole stretch, after splicing, these salmon-colored parts that got made into an RNA, the hnRNA, the pre-mRNA, they all got transcribed, but they get cut out. And ribosomes only see the stuff that's shown in the dark red, the 5' untranslated region, the 3' untranslated region, and the coding regions in between those UTRs. This is huge. The excision of those intervening sequences, more than anything, is responsible for why eukaryotes and humans in particular are as complicated an organism as we are and have the capability that comes from that complexity that we've got. If not for splicing like this, we'd be in a world of hurt. We'd have a much harder time dealing with the challenges that come to being a complicated organism if you couldn't pull off splicing. It's a big deal. And I don't think I can impress upon you enough at this stage in this class how big a deal this is. But I'm telling you, boy, I am a believer. I accept this is a big deal, all right? And so you'll be persuaded, you'll be brought into the fold in subsequent classes. But for now, just tolerate me, you know, go with the flow. The guy thinks it's a big deal. All right, it's a big deal. Splicing is a big deal. Let me tell you some of the things that makes this a big deal. I've already told you that it is splicing that allows one gene at the DNA level to code for multiple proteins, right? Nothing I've told you so far explains how that happens. But there's a simple extra thing I can tell you. What gets recognized as a region that gets cut out from the pre-messenger RNA can be different from one kind of cell to another kind of cell. In a word or two, splicing can be tissue-specific. So muscle cells in the heart might recognize something as an intron that muscle cells in your quadriceps don't think is an intron. Contemplate that for a little bit, all right? At the level of DNA, it's all the same, but in your heart cells, you might cut something out that you left in your non-heart muscle cells. What happens? What happens is that the same gene is going to give you proteins with different amino acid sequences, a lot of stretches that may be very similar, but ultimately with different amino acid sequence in one tissue type as opposed to another. All right? Any of you guys remember Dick Cheney? He was Bush's vice president. Bush didn't get a lot of respect for being smart, all right? I think history will, whether he was smart or not is a separate issue, but people didn't think he was smart, all right? There was a lot of talk that he wasn't that bright. And so there was concern when Dick Cheney, who had a history of heart problems, looked like he might have had a heart attack because people were thinking he's the brains behind what's going on in the White House here. And if we lose Dick Cheney, we got problems because we got a guy who's a dim bulb who's going to be calling the shots. So people were worried when it looked like Dick Cheney may have had a heart attack. All right? Well, at one point, Dick Cheney got rushed to the hospital, and people are thinking, oh no, he's had a heart attack. And people, but the White House press corps, the press secretary came out very quickly and said, we don't think he's had a heart attack. He's fine. He's going to be back to work in a few hours, should be all right. And then, like a day later, the press secretary said, well, it turns out that the Vice President Cheney did have a mild heart attack. And people were saying, oh, you slimy people. You knew he had a heart attack all along, but you were just trying to make us feel good and tell us he didn't have a heart attack. And now that he's better, you're telling us he really did have a heart attack. Why did you lie to us? What was going on? They weren't lying when they said they didn't think Dick Cheney had a heart attack and then later on thought that he did. You know what happened? It took that long for them to get molecular proof that he had had a heart attack. And you know what that molecular proof was? The protein myosin in heart muscles is coded for by the same gene as the protein myosin in your regular muscles. But because of alternative splicing, cardiac myosin has a stretch of different amino acids than regular myosin. And it took a while. You know what happens in a heart attack? The definition of a heart attack is that some of your heart cells have died, right? They get killed. It took a while for those heart cells to get so dead, if there is such a thing, that they busted open and let some of that cardiac myosin get out into Dick Cheney's bloodstream. And they were monitoring his blood to see, is there any cardiac myosin present? That whole test, if you know somebody who's had a heart attack, they assess how bad your heart attack was by how much of an increase in cardiac myosin there is in your bloodstream. All right? And you couldn't do that if not for the fact of alternative splicing. So that's one thing, right? There's a medical application that comes from this alternative splicing. But you wouldn't have a functioning heart if your heart's myosin wasn't different at the amino acid level from your quadriceps myosin. And so much more important, I suppose, is that. But so there's alternative splicing for you. That's one reason it's a big deal. There's one gene, the record holder within the human genome for the most number of introns. One of our genes, I think it's human growth factor 8, something like that, as I recall. 118 introns. How about that? So you transcribe this huge monster of a gene, and you cut out 118 chunks of it and link together the exons before a ribosome ever sees it. How about that? That's huge. It's a rare gene. There's just a handful that we know of within the human genome that do not have at least one intron. Most genes have 2, 3, 4, 5, 6, something like that. One outlier has 118. You guys have heard of cystic fibrosis, right? Nasty genetic disease. The protein that's got an issue for people who have cystic fibrosis is CFTR, cystic fibrosis transmembrane regulator, right? CFTR. It's a big protein as far as proteins go. It's about 3,000 amino acids long. Most proteins are just a couple hundred amino acids long. This is a big boy, 3,000 some amino acids long. Do some math, right? Let me challenge you to do some math. How big do you expect the cystic fibrosis gene to be? At a minimum, right? I told you it's 3,000 amino acids long. How many nucleotides long must the gene be? Simple math. Each amino acid is coded for by a triplet codon, 3 times 3,000. At a minimum, the cystic fibrosis gene's got to be 9,000 nucleotides long, right? I don't mean to scare you with that math. This is easy, right? At a minimum, and give it the 5' UTR and the 3' UTR, okay, 10,000 nucleotides long, right? On paper, that makes sense. You can see where I'm going. I'm going to drop a bomb on you right now. You know how big the cystic fibrosis gene in the human genome is? Brace yourself, right? It's bigger than 10,000. You saw that coming, I'm sure. You know how big it is? 3.2 million nucleotides long. 3.2 million nucleotides long. The messenger RNA is 0.01 million nucleotides long. You transcribe, you make an RNA copy of something that's 3 million nucleotides long, and you throw most of that stuff away to cobble together a messenger RNA that will make a 3,000 amino acid long protein. Splicing is a big deal. How big is E. coli's genome? You remember? About 4 million nucleotides long. I just told you, you've got a gene that's given the whole E. coli genome a run for its money in terms of its size, and yet you throw away most of that information on their way to making a messenger RNA that gets used to make that protein. Eukaryotes do this. Prokaryotes don't. Splicing, it's a big deal, right? For a whole bunch of reasons, and I hope that I've impressed you with some of the cooler trivia associated with splicing. But that's what's going on with splicing. And I'll tell you now, not only is it a big deal, it's tough. It's hard to pull this off. Because, accept this please, it has to be done spot on, correctly, every time. There's no partial credit that nature gives when it comes to splicing. It's not good enough to have spliced out 117 of the 118 introns of that record-holding gene. You got to get every intron out for it to be a useful messenger RNA. And you got to link up the exons exactly the right way. You can't be off by a nucleotide or two. And I'll show you why in a couple of slides. But here is how that splicing is taking place, sort of a graphical representation. It also lets me tell you about another interesting idea. So here we've got a gene at the DNA level. It's got 3 exons. Those are the things that get linked together that ribosomes are ultimately going to see. And if you've got 3 exons, that means there's introns in between them. If you've got 3 exons, that means 2 introns. They give you the names for the exons here, curiously. Exon 1, exon 2, exon 3. The introns get names too, by the way. Intron 1, intron 2, even though they haven't bothered to show that for you in this picture. But you transcribe the whole thing, you splice out the 2 introns, you link together the 3 exons, and that's what gets translated. What they're trying to show you here with this picture, what I guess I'm wanting to convey with this picture, is that very often we find that introns correspond to functional units within the protein. So you might have a protein whose job it is to bind ATP, right? We got a string of amino acids that binds ATP. But binding ATP, that's kind of lame. You got to do something with the ATP. How about if you not just bind ATP, if you also bind to some other protein? You can recognize another protein. Because now what you can do is you can have this protein both bind ATP and bring it so that it's in close proximity to another protein. And then let's put on another domain, another functional unit that lets you take a phosphate group off of that ATP and transfer it to that protein that you've bound to. This is a winner. Now we got a protein that can do something and do something useful. 3 separate jobs all pulled together into one protein. Each of those jobs accomplished by a different chunk, a different region of the protein. It turns out those chunks, we call them domains, those chunks often correspond to where you find intron-exon boundaries. An exon often corresponds to a functional domain for a protein. And the stuff in between often corresponds to nothing. So anyway, you can piece together proteins with multiple functional domains. The way the splicing happens, again, it's a big deal. Lots of study into this. The short answer, the cliche answer is, how do you do splicing? A protein does it, right? It turns out it's a bunch of proteins does it, or do it, come together. A bunch of proteins and some RNA molecules come together to make things called spliceosomes. It's spliceosomes that are responsible for recognizing the beginning of introns and the ends of introns, and then bringing together the 2 exons, breaking the phosphodiester bonds here, and linking together, making a new phosphodiester bond between the end of one exon and the beginning of the next exon. You end up cutting out the intron, and you leave behind a spliced messenger RNA. So the one new thing here, perhaps, for you to focus on is that it is spliceosomes. That's the thing that's responsible for doing this splicing business during RNA processing. All right. So, time to talk a little bit about protein synthesis. I don't know, I guess that looks a little scary, right? It's not too bad when you stop and think about what's going on here. This is a ribosome, okay? Ribosomes come in two parts, both in prokaryotes and eukaryotes. There's two parts. There's a large subunit and a small subunit. Aptly named, right? Guess which one's bigger? You don't need me to tell you. You can figure that one out on your own. You got a large subunit and you got a small subunit. Those two subunits come together and an RNA molecule snakes through it in the middle. Come back. Well, at least I get the picture on the screen. Don't seem to be able to advance the slide. Oh, now the picture's off the screen too. So now we're in good shape all around here. Well, that's interesting. Haley, do you think you can talk some sense into this? Oh, here, this might do it. Just start fresh. All right, well, I don't know. I pressed a button, apparently. I'm a professional. I've done these things before. You know, as a graduate student, when I was a graduate student, I got to attend scientific meetings for free. I had a marketable skill. And my skill was I was the projectionist. And I'd sit in the back of the room by the projector. And to demonstrate your prowess as a projectionist, these are in the days when they had these things called slides, little pieces of plastic, or even in the old days, glass, right? But you'd demonstrate your prowess by being able, like if a slide was inserted upside down or backwards or something like that, you would be able to take the lid off the little thing and flip the slide around. You based on what you saw, you'd flip the slide around. Like the faster you could do that, the better of the projectionist you were. So, you know, I have those skills, and unfortunately, they're absolutely useless these days. But I could kick that projector, and maybe that would help us. That was one of the things that was a fail-safe. We also learned how to change the light bulbs in the projectors. That also is something that's a useless skill today. But check it out while you still can, and the picture's still up there. There's an RNA molecule snaking through this ribosome. Notice the scale, right? The RNA in the individual nucleotides are puny compared to this big old ribosome, the large subunit and the small subunit together. There's an interesting matter of scale here. You guys may have actually seen ribosomes with the light microscopes that we have in the freshman biology lab, right? That's a big molecule. If you can see it with a light microscope, you got no chance of seeing a messenger RNA molecule with a light microscope. You might be able, if you're lucky and you look hard and you trick yourself into believing, you might see a ribosome, okay? Difference in scale. The ribosome moves along the RNA from the 5' end to the 3' end. And as it's moving along, it's got within the ribosome a spot to hold 3 tRNA molecules. There's the tRNA molecule that has just been used. There's the tRNA molecule that is checking out a triplet codon at that instant. And then there's the tRNA molecule that's looking ahead in terms of trying to figure out what the next amino acid, what the next triplet codon is likely to be. The tRNA molecules bring with them amino acids that can get strung together then to make a new polypeptide that snakes out through the exit tunnel. I prefer to call it the butt of the ribosome. That's my lingo, right? You guys will probably want to be, I'm a full professor. I can say stuff like that. Nobody bats, oh, he's eccentric, he's a full professor. You guys should refer to it as the exit tunnel, all right? And actually, you might want to use that phrase exit tunnel in other contexts too, you know, sound more sophisticated in some of your other discussions perhaps. But the protein comes out the exit tunnel as it's being synthesized. So there's the general picture of what's going on. Here we go again. Maybe it's that slide that's the problem. Here is a less filled-in version of what's happening. You can see you got the ribosome chugging along. It's moving in the 5' to 3' direction. Here's the 5' end, there's the 3' end, and it's bringing in, it's got room to hold on to 3 different tRNA molecules. Here the tRNA is shown as this sort of bent rod kind of thing. Check out what these tRNA molecules are doing. At one end of the tRNA molecule is a thing called the anticodon. Right? 3 nucleotides that can base pair specifically with the 3 nucleotides of the codon. All right? We've been talking about codons a bunch now on Tuesday and today. Here's a codon. It's UUU. Do you know that one by heart yet? So you know AUG is the start codon. If somebody really pushes you and asks you, tell me another one of the 64 triplet codes and the amino acid it corresponds to, UUU is the one in the upper left-hand corner of the genetic code, right? Phenylalanine. I happen to know that one by heart because it's the one in the upper left-hand corner. But at the RNA level, you've got a UUU, 5' to 3', that will base pair specifically with something, a tRNA that has an AAA in its anticodon. There's only one tRNA that has AAA as its anticodon. It's the tRNA to which phenylalanine gets attached. When this tRNA finds its complementary codon in this RNA, it brings a phenylalanine to the ribosome party, and it gives that phenylalanine to the ribosome to add to the growing polypeptide string. Once that tRNA has done its thing, it gets kicked to the curb, and the next tRNA, which has already been scouting out what it might base pair with, the next tRNA gets its moment in the spotlight, and it brings in its amino acid, and it gets added to the chain, and you continue with that extension. For this to work, you have to have some ability to cycle through tRNAs. If this one's not looking too hot, right, I need another one in there fast. As fast as you can get me one, the better, right? A particular tRNA, you bring in amino acid protein synthesis happens at a clip of about one amino acid for 1/10 of a second. So you sort through all the tRNAs. I know it's gone. Right? You sort through all the tRNA. It's back. Right? You sort through all the tRNAs. Dozens, scores, maybe hundreds per second, so as to make sure you got the right tRNA. When you have it sitting there for a fraction of a second, because the base pairing is right, the ribosome snatches the amino acid off, and you move on to the next codon. And you always go 5' to 3' to do that. So before you saw the tRNA as this sort of bent rod, this is what a tRNA looks like in all its glory. Okay? And I'll tell you, I was surprised. Like 2 years ago when I was teaching this class, I flashed a picture of a tRNA molecule up on the screen before we started talking about this stuff. How many of you guys know what this is? Have you ever seen anything that looks like this before? Nobody. And they were probably all just shy. But nobody in a class of a couple hundred people ponied up the idea that that structure on the left side of the screen is a tRNA. And that kind of boggles my mind. You guys know in kindergarten that DNA stands for deoxyribonucleic acid, but nobody ever bothered to show you this structure before. tRNA molecules, at least when I was going to school, this was an iconic sort of structure. And there's still some neat stuff that you can see sometimes. There we are, in this tRNA molecule. So all tRNA molecules have this basic structure. All of them look like this at a molecular level. And they have some features. All tRNAs have some features in common. They've got this thing called an anticodon loop, right? Good name, right? That looks sort of like a loop, right? Some people refer to this as a hairpin loop. Guys, you know what I'm talking about? I bet you the women here know what I'm talking about with a hairpin. Maybe a bobby pin, would be another way to think of it. But this is a hairpin loop. And it's specifically, this one is the anticodon loop, because what's going on, those are the 3 nucleotides that are going to be base pairing with the codon. That's the anti-codon that base pairs with the codon. There are two other hairpin loops on all tRNA molecules. All of them have this, all right? And all of them have at their 3' end, and notice it's an RNA molecule, so instead of T's, you see U's, but like any nucleotide or polynucleotide, it's going to have a 5' end. This is the beginning, and this is the end. It's got a 5' end and a 3' end, and it's to the 3' end that the specific amino acids get attached. This anticodon means you get one specific amino acid attached over here. If you had a different anticodon, you'd be attaching a different amino acid up here. All right? So this is the structure. And when you're looking at this structure, what sorts of things do you see? Some things that are kind of interesting, I hope, right? It's got a 5' end and a 3' end. It's got a beginning, it's got an end. It's got an anticodon, right? It's got, here's a fancy word for you, intramolecular base pairing going on. You guys probably always have thought, if you've ever thought about them, is RNA molecules is single-stranded molecules. This is an RNA molecule. Yeah, it's got some single-stranded stuff in it, but a big part of the functionality comes from the fact that this RNA molecule folds up and sticks to itself with intramolecular base pairing. There are hydrogen bonds that are forming between the nucleotides at the beginning and the nucleotides at the end. And from this part of this hairpin loop and this part of this hairpin loop, notice how they're trying to help you out here. Where there's a C, you can base pair with a G. Where there's a U, you can't base pair with a G, but where there's a U, you can base pair with an A, right? This is a stable structure. Energetically, it's stable. Those hydrogen bonds, when you can see them, will tell you this is sticky, and it likes to fold up in this way. And one more time, it's not the last, but one more time, I can tell you that in biology, structure and function are interrelated and a big deal. It's the fact that this tRNA molecule folds up. Now it's just playing with me, you know? It's the fact that it folds up in a particular way. Oh, I made it really mad by saying it was playing with me. It's the fact that it folds up in a particular way that allows the tRNA to actually do the function, for ribosomes to be able to latch onto it, fit it into the right spot, and so forth. So the structure and the function of those tRNAs are important. I appreciate that the projector is working. Let's see if that helps. All right, so pop quiz kind of time, right? You got a model of a tRNA here. I'd love it if I had the time to put a graphic like this onto your final exam. You know, if you want me to be a happy guy, you'll root for me to have an extra 5 minutes so that I can copy and paste this picture onto your final exam. I've yet to have the time, but one of these days it's going to happen. And if you happen to see it, smile, because you'll know that I was happy that I finally got to put the picture on the test. I'd love to put a picture of this tRNA molecule on your final exam and ask you stuff like, where do you see the 5' end? Well, here's the 5' end. Where's the 3' end? Over there. Where are the hydrogen bonds? Oh, look at that. Those are hydrogen bonds, right? Where are some regions that aren't paired? Well, there's no base pairing going on there. Where's the anticodon loop? There it is. Where's the binding site for the amino acid? It's up there. Right, so these are all structural features of a tRNA. Messenger RNAs get a lot of attention. tRNAs don't get paid a lot of attention. For each of the 20 different amino acids, there is a specific tRNA. That's what those tRNAs look like. All of them look like that. They differ in terms of what their anticodon is, as well as a few other nucleotide sequences along the way. But tRNAs, that's the iconic image of a tRNA. Okay, well, we're in the home stretch. We've talked about translation now as much as I care to, and ribosomes and how they're elongating a growing polypeptide, amino acid at a time. All right, let's talk a little bit about mutations, and I think we'll call it a day. So on Tuesday, I talked with you about sickle cell anemia. I told you that at the heart of it all was a single nucleotide difference in one of the two proteins that get used to make the protein hemoglobin. In people who don't have sickle cell anemia, at one point along the way in that gene, they've got a codon that is GAA. GAA codes for glutamic acid. People with sickle cell anemia have a different codon. They've undergone a mutation. They've got GUA. GUA codes for valine. This is a non-synonymous substitution. Okay? Let's break that down. Substitution. Let's start there. We've substituted an A for a U, right? That's the substitution. It's non-synonymous because that substitution at the DNA level is resulting in the use of a different amino acid than you would have had otherwise. A synonymous substitution, in contrast, is one where you change the nucleotide sequence at the DNA level, but it doesn't change the amino acid at the protein level. And again, you can do that because of the redundancy of our genetic code. Leucine, for instance, has 6 codons that are synonymous. If you change the nucleotides to give you any of those 6, and you started with one of the 6, you've made a synonymous substitution. But this is a non-synonymous substitution. And as I was telling you on Tuesday, that in turn gives rise, it's solely responsible for the phenotype, the disease state that we call sickle cell anemia. One nucleotide change, one amino acid substitution, and one protein, and you can have a problem like sickle cell anemia. So let's talk about other kinds of substitutions and see how these systems can get messed up, because people have bothered to take the time to give each of these different kinds of problems a name. So let's say this is the sequence of a gene in people who don't have a condition. The RNA that you make from it looks like this, and it codes AUG. Remember that one, right? That's the one that I think you might want to actually remember. That's the start codon. The first amino acid in the polypeptide is methionine. There's your start codon. There it is in all its glory at the DNA level. And then you look at the next set of 3. And I don't think I can emphasize enough. You look at the next set of 3, right? You found your start codon. That establishes a register. Everything else has to be 3 at a time after that. All right? You got to look at these 3 at a time. So you got a codon that says AUG. The next 3 are AAG. Apparently that codes for lysine. The next 3 are UUU. We know that one, right? That one is phenylalanine. The next 3 are GGC. Apparently that codes for glycine. Triplet codon, amino acid. Triplet codon, amino acid. And then this is a really short protein, frankly so short that it can't do anything of consequence, and I mean that seriously. We'll talk about this more later on Tuesday of the week after Thanksgiving. But here's a stop codon. There is no amino acid that it's coding for. That tells the ribosome stop translation here. So you've made a 4 amino acid long peptide, a quadripeptide, tetrapeptide, I think we would call it. All right, not that big a deal. But look what could happen. You could have a substitution, right? You could change this, what was it? Instead of the last one there, instead of a G and a C, you could change that to an A and a T. That's different, right? That changes the codon. The codon used to be GGC, now it's GGU. You would think that was going to mess stuff up, wouldn't it? You've made a mutation. It turns out this is a silent mutation. It's a synonymous substitution because we've just taken one of the codons for glycine and we've replaced it with the other. In terms of the functionality of this polypeptide, if it had a function, you wouldn't expect there to be any significant change. The amino acid sequence is the same. How could there be a difference, right? So some substitutions are silent substitutions. They give rise to synonymous amino acids. Of course, there's going to be other kinds of substitutions that change the amino acids. A non-synonymous substitution, we could call that a missense substitution. Here, instead of changing the third nucleotide in the codon, the first nucleotide in the codon is changed. Instead of GGC, it's now AGC. That is a non-synonymous substitution, a missense mutation. Instead of having methionine, lysine, phenylalanine, glycine, we've got something different, methionine, lysine, phenylalanine, serine. You would expect that has greater potential to cause a problem. So we've talked now about missense mutations. Open up your minds to the idea of a nonsense mutation. Missense makes sense. You've misinterpreted. You've put in the wrong amino acid. Nonsense effectively means you've put in no amino acid. You've introduced a premature stop codon to the sequence. All right, so here, instead of AAG, what you got is a change that causes you to have TAG. In TAG, at the RNA level is UAG, that's a stop codon. So if you thought that changing the glycine for serine could cause a problem, you'll be very easy to persuade that truncating the protein so that it's only one amino acid long, that's almost got to cause a serious problem. So missense mutations change an individual amino acid. Nonsense mutations introduce a stop codon sooner than the real stop codon, and you truncate the protein, you make nothing beyond it. In the scheme of things, nonsense mutations are almost always a whole lot more consequential than missense mutations, for those obvious reasons. Consider this one here. So I've tried to say a couple times now, you read those triplet codons 3 at a time carefully. The ribosome can't slip a little bit, one nucleotide forward or back. If you have a mutation that doesn't change the nucleotide, but rather adds a nucleotide or two, you get out of register for that reading frame that the ribosome would be looking at. And in the moments that we have before the screen turns off again, notice that, wait, what's happening? So we've got here a nucleotide insertion at the beginning of this open reading frame that causes us to have a stop codon at the beginning that you wouldn't have had if not for this extra nucleotide getting added. Fancy word for that kind of change. We've got missense, we've got nonsense. Now we can start talking about a frame shift. The reading frame has been shifted. Sometimes frame shift mutations will introduce a stop codon. Even in those circumstances where they don't introduce a stop codon, they change the reading frame, and pretty much all the amino acids downstream end up being different because you end up being out of register. I tell you, we need to have somebody figure out why this computer's not working. So frame shift mutations can be when you add a nucleotide or 2, or 4, or 5, or when you delete a nucleotide or 2, or 4, or 5. Did you notice I didn't say 3, right? 3 isn't as big a problem as 1 or 2 or 4 or 5. 3, at least all the rest of the amino acids will still be the same as if the deletion or insertion hadn't occurred because you've taken out a whole codon. So frame shift mutations tend to be a pretty big problem. All right. Well, I tell you what, we got 2 minutes. So what I want to do is I know I'm not going to see some of you for 10 days? Tuesday of next week. What's going to happen on Tuesday of next week is I'm going to talk with you about how viruses pull all this stuff together to do their thing. That's kind of cool. But then when we come back the following Tuesday after Thanksgiving, I am going to reveal to you the wonder that is the lactose operon. And even if you've talked about the lactose operon in high school before, I'm going to tell you stuff about the lactose operon that's going to blow you away you've never heard of before. And if I'm on a roll and making good time, I'm going to tell you about the tryptophan operon and tell you how it is that prokaryotes are able to turn genes on to make proteins only when it's to their best advantage. It's an amazing system. So that's what you have to look forward to after Thanksgiving. Between now and then, I'll tell you about viruses, and we'll all eat some turkey. All right? Have a great weekend. See some of you next week. See the rest of you in 2 weeks.